I am Manolis DIALYNAS. I am a member of the Bioinformatics support group here at IMBB. The group was founded about a year ago, and its purpose is to serve all the personnel and the PIs of the Institute and collaborators and partners, and to help them with their bioinformatics problems, or give them some hints or help in general. Bioinformatics is not a new field. It's very, very old. Initially, all the software and the information, the databases and everything was proprietary. Users had to pay licenses for it. In the last 20 years, there seems to be a shift towards open source. I'm mostly referring to biology, okay, not biomedical. Okay, biomedicine is it is a little bit different. You still have to pay lots of money to get data and tools. And also, as everything else, there's been a shift also from command line tools, that applications that users had to install on their computers and use command line to run programs and get data. We now have moved to the web, which means that it's not something different from what we had before. It's just we have a much easier interface now. Okay, so users are more compelling to use the web interfaces than command lines. Okay, so we are at the web now, and what can we do here? There's a myriad of resources available. Okay, this is a very draft categorization of the resources available. We start with repositories and databases, where people can submit and retrieve plain data such as sequences or experimental data. The next step is some sites give you the option for data mining, which is to answer some questions in the form of queries and get some of this data that are relevant to what you're doing. Okay? So, for example, in a repository, we have billions of sequences. Okay, if you're working with Drosophila, you're only interested in the Drosophila sequences. So, a very simple query is to get only the sequences for Drosophila. Okay, as soon as you get the sequences, you have to do something with it. I mean, analyze it. Maybe combine it with your results, and hopefully come to some conclusions. Okay. Next thing is sharing. If you just keep it to yourself, then there is no progress in science, of course. So there are sites that help you share the information and the data from your experiments. Collaboration also is very important. For example, if you want to annotate a whole genome, yes, even if you have your own lab and you do all the work at your lab, the people in your lab have to collaborate. Okay, it's not possible for all of them to sit in front of a single computer and do the work. So if you have collaborators around the world, everybody simultaneously can help you in your project. Okay, and some of these sites also give you what is known as an application programming interface, or API. This is a programmatic way to access and analyze the data locally. So if you have a software developer in your group, they will develop an application that will go on one of these sites, get the data, do the analysis that you want, which can be analysis algorithms that are already known, or your own algorithms, and come back with some, hopefully with some user useful results. Also, a very important thing about the web is that it allows you to access high-performance computers. Okay? If you were to do this, all this in your lab, then probably you could have a server, one or two, and do the work there. Okay? But the size of today's data, it's very, very large. Okay? You need very fast computers to do the job. And these are computers that cannot be housed in a single room. Okay? So moving on, and actually starting the presentation, it would have been a heresy to start it without mentioning these three organizations that have formed a consortium, which is called the International Nucleotide Sequence Database Collaboration. Okay, this all started about 35 years ago, and probably this doesn't tell you much, but probably you all know Genome. This is the Japanese and the European partners. They all have the same data. It's scattered around the globe, synchronized daily. And what you can do here is, this is a web interface for ENA. You can just come here and fill in some keywords of the gene that you are interested at, or a partial sequence. And this is what you're going to get. This is a summary. In this one, we got more than 6,000 results. And let's say we're interested in the mosquito cool excipients. So we click on this, and we get this screen. This gives us some information about the species. Here is a primitive. This is a primitive because you're going to see later a much more detailed visual representation of your gene. And down here we have the complete sequence of the gene. And if we click on this tab, we're going to get some references for the people in the study who submitted this sequence. So this is this dates back to probably 1982. Up to 1990, this is probably was your only easily accessible resource. Since then, we moved on. Ensemble, it's a European organization. It belongs to the European Bioinformatics Institute. Was the next big step. It has introduced many features, mainly visualization and easier analysis. These include the Genome Browser, gene expression visualization, variants. Okay, it's much easier now to visualize various forms of your genes, and also it introduced a cross-species comparison. This is the main page of Ensemble. You can see all the tools that they have available here. It would take not hours, but days just to go through Ensemble. Okay, it's a huge resource, very useful. Right now, they have more than 50 species available in here, and I'm just gonna go briefly some highlights. This is the Genome Browser Ensemble. It looks a little bit the same as the one we saw earlier, but if you see here, they have started doing some more work, and you can get some more information. For example, here you can see promoter regions, exons, introns, and you can see actually the transcripts. And in this example, we have two transcripts, this one and this one. This interface is packed with data. This is a very, very plain form, but there are lots of options here. You can configure your page if you want to select the data that you're going to want to look at. And also, almost everything on this map is clickable, and pop-up windows will come up and show you more information. So in this one, up here we're looking at the forward strand. We can see the locations on the chromosome. And if you click, you can get more information here. You can get the precise coordinates up here. And base pair and amino acids. All these lines are called tracks. And if you want to configure this page, you can visualize your own tracks. For example, if you have your own annotations, or if you have come up with a different transcript, you can ask an ensemble to connect to your data server, get the data, and visualize everything in here. Moving on, this is a more detailed view of exactly the same gene that we were looking yesterday. In here, I have asked Ensemble to give me some more details about the structure of the protein that arises from these transcripts. And here it has identified some signatures from different other resources. SMART, PFAM, and Interflow is not here. I'll show you later. So this was the second step. Moving on, if we want to get more information, then we have to visit more specific databases. Okay, and these are Organisms-centric database. Flybase for Drosophila, SGD for Saccharomyces, C. elegans, Mus musculus, Corm, Plasmodium, and vector-base, all the insects.
Humans?
Human, we stick to Ensemble.
Okay.
Since I'm more familiar with vector-base, because I worked for it for 10 years, I will move on to vector-base, which uses the Ensemble Genome Browser, but it has a few additions. You can see here they have gene ontology terms for the genes, and many other annotations and data like SNPs. We're still looking at Go annotations here for the same gene, and this is a more detailed view with all the variations that have been reported and deposited to vector-base. Very nice displays, very useful, but packed with data. Every one of these little lines is clickable, and as you click on it, you're going to get something like this. So here we're looking about SNPs. It tells you what has changed, and what is the consequence of this change, and the evidence. There's no evidence here, just the source for this. Okay, this is again from the Ensemble Genome Browser, but within vector-base. Instead of gene view, we have now switched to the transcript view. Okay, and here they have done some analysis, and they have come up with some signatures for this peptide. What could be the potential product of this peptide? Okay, so this gives sodium ion transport, voltage gated, and some structural pieces of that transcript, the protein that will result from this transcript, that seem to be a voltage-dependent channel. Okay? Here's a very plain calculation of the peptide, isoelectric point, molecular weight, and number of residues. If you want more information about this peptide, then usually you can move on to UniProt, which has data on manually annotated and reviewed peptides, but also on electronically analyzed and annotated peptides. Again, UniProt is a huge resource. Very briefly, what we can do here is come to the Ensemble Genome Browser, select the peptide we want, get its sequence, copy the sequence or part of it or whatever, bring it in here. This is the Interprobe website. Click on search, and this is what we're going to get. Okay? So from the sequence, we went something it's a little bit better visualized. Again, we have ontology terms here and some structural information about our peptide. Moving on, some PFAM signatures that we can find in our peptide, and this will give us an idea of what our protein does. Very brief one, a generic one. Moving on, again, I got the same sequence that we used in UniProt, and we came to this site, which is called Predict Protein. This is the open version of it. Again, we paste our sequence, and we come up with which looks something like this. This is the website. It took about 20 minutes for this supercomputer to come up with these results. This is definitely something that you cannot do on your desktop. So, as you can see here, pretty much everything on this map is also clickable, or if you just mouse over, it's going to give you more information. So this is all our peptide, and here we have a protein binding region, and here in this position we have a polynucleotide binding region. You can see down here some structural information about this peptide. That there is a helix here, another helix here, and if we move on to the next track, the track just below it tells us where this portion of the peptide is exposed or buried within the protein.
But do you actually have 3D structural modeling? I mean,
not yet. Yes, you can do it. But
the group, like my support group, is they doing it or not? I mean, if someone wants to have a 3D structure and play with the protein structure and see the amino acids that are exposed, vary, other ligands, and do this game of the protein. Are you able to do it, or?
No, in this, you have to come in contact probably with Dr. Korkinidis in his lab. And first, you have to get the peptide, crystallize it, and then do all the rest, right? Okay. So we're talking about just computational analysis of the peptide.
Like viewer, for example.
Yes, I will mention it briefly later.
But if one has a problem and wants actually a 3D structure, will you be able to do it or not?
PDB viewer is only to display proteins that are already available the sequence and the coordinates of the molecules are
Yes,
already reported in PDB.
Then
Viewing it is simply. You can
then make changes on that, the mutations, add the ligand, and see.
Then you have to recalculate the structure if you add a ligand or whatever. And at that point, all the prediction software for 3D structure are merely predictions.
Yes,
So,
talking about predictions.
yes, yes, there is a battery of software to predict 3D structure. Maybe Kyriakos is more expert than me to tell you that those predictions are not quite right. So I don't know how trustable they are.
We have used these predictions a lot in the lab.
Here's the
Mine
success
doesn't
and
work.
success.
But yes, in the range
The
that
one that works with proteins, this is very, very essential tool.
yeah, in the range that we cannot go further on what the model or the software predicts. Yes, we can run such a prediction and to do not guarantee any success or whatever on this. So yeah, exactly. We'll run. We have there are some pipelines. We can run such a pipeline. We can give you the result, but there is no way to check it or to be confident that this is it.
What is the
I wanted to ask that here in this question, it says evidence prediction. So it is predicted according to the sequence display of this area that it does, it is a variant or it interacts with a protein or anything.
There is no hard experimental data that supports this.
I wanted to, so if there is a publication that supports that this domain is a protein binding domain, it is not.
There's going to be a citation for it.
There it goes.
Usually, if you see something it's more relevant in Ensemble, it says it has the acronym IEA, which means inferred by electronic annotation. So this is just plain computational. It's based on homology, probably. If they have identified this very similar protein, this is they're going to use it as a model.
Have they run these predictions through the test to see what is the relation of this to the real world?
It's supposed to be the best that we can get right now.
Yeah, sure. Sure. I'm sure that they'll put something worse than what they can get.
On the next screen, they even have some scores about how, okay, it's about genotology terms, but they're pretty good. They're pretty good. I'm probably not going to tell you the whole truth about the full peptide, but for some regions, it's pretty accurate. And again, it's not because they're doing some magic, but it's because they're based on homology from known peptides. Another on the last track here, it can be very useful. For example, if you want to design, if you want to create an antibody against your peptide, okay, it tells you which regions of the peptide are exposed or buried within the peptide. Or in this example, it tells you if it's that this is a helical transmembrane region, and if this is what you have selected to be the epitope of your antibody, probably it's not going to work in the real world. Is this clear? Okay, because this portion of the peptides is actually in the membrane. So if you want an antibody for this peptide, your antibody will probably never stick to it. And down here, they have all the proteins that have sequence similarity to the ones that you're looking for. Okay? So
Up to what degree of similarity, or is it really similarity or identity?
it gives you some.
So do you have like a threshold or?
That They give you all this information right here. Okay, this is the sequence that we searched for was from cool experiments, a mosquito, and this one is from Anopheles gambia, another mosquito. And this one, also from Anopheles gambia. Probably they're homologous proteins, all of these. Okay? Just show you one more. Again, genome ontology terms with Q. And at this time, we'll have a reliability score here. Okay? A very quick look, they can just tell you what probably your gene does and to what degree of confidence. Binding sites. If you just want to focus on the binding sites, protein binding sites, and nucleotide binding sites. And probably this is wrong, okay, because this is a sodium channel, voltage-gated sodium channel. I don't know if there should be a polynucleotide binding site on it. Okay, moving on. This. If you have, if you're in the mood, you can do your own gene ontology annotation. And as Pandelis had mentioned in his seminar, David is the application that you want to use for this. It's pretty straightforward. It's just a pipeline which is very detailed in this paper up here. You just get your data, you come here and you feed in your data and hopefully get something meaningful. The other thing you can do is visit KEGG, again submit your sequence and get some more helpful information from there. And if you want to move on to something more fancy, let's put it that way, there is a platform that's called Galaxy. It's very popular, has a very active community. There are about 70 public servers available throughout the world. We have recently installed one here, and this is what we can do. Okay? Next generation sequence experiment analysis. It's very flexible. It allows you to design your own workflow. You can do whole genome annotation, and there are some plugins in Galaxy that will do many, many things. One of these is RNA structure prediction, and you can go even further, okay, to protein structure prediction. The other platform that we have installed here and we have started using it is called Chipster. We use it to analyze next generation sequencing data and microarrays. It has a fairly simple genome browser. You can do sequence alignment, pick up and find your variants. And for RNA-seq experiment, you can do assembly and calculate a differential expression. And for ChIP-seq, peak detection and motif scanning. So there should be and here. If you want to analyze microarray data, what we can do is normalize, filter, draw some really nice charts, very helpful, very informative charts, do clustering and pathway analysis. And this is what chipset looks like and what you can get out of it. Okay, this is a Genome Browser, multiple reads, SNP detection here. This one is an expression profile, some genes here, and this one is clustering of the genes right here. Okay, again, this talk by no intention was to be a very in-depth presentation, okay? It was a very, very brief overview of what's available there, how things have evolved, and what we, the Bioinformatics Support Group at IMBB, can help you with. So please, if you have any questions or you want help with something, contact us. And these are the links for the websites that you saw earlier. Okay, thank you.